Good day, guys. We'll be having an extensive lecture on the neuroglia, also known as the glass cells. And before we start, just for you to know that the neuroglia are the supportive cells. They are not really neurons. Okay, they are not neurons. So I'll be making a video to differentiate between neuroglia and neurons very soon. So neuroglia, also known as the glass cells, these are the various examples of neuroglia. We have the oligodendrocytes. We have the ependymal cells, we have the strand cells, we have the astrocyte, we have the microglia. Okay. The neuroglia, also referred to as the glass cells or simply glia, are non-neuronal cells that maintain homeostasis. Okay, they form myelin and provide support and protection for the neurons in the brain and the peripheral nervous system. So the main functions of the glial cells include what? To surround the neurons and hold them in place, to supply nutrients and oxygen to neurons, to insulate one neuron from another, to destroy pathogen and remove dead neurons, and regulation of internal environment of the brain. Now, during early embryogenesis, glial cells direct migration of neurons and produce molecules that modify growth of axons and dendrites. Now, recent research indicates that glial cells of the hippocampus and the cerebellum participate in synaptic transmission, regulate the clearance of neurotransmitters from the synaptic cleft, and release glial transmitters such as ATP, which modulates synaptic function. Okay, and glial cells also function to speed up velocity of conduction in nerve fibers. Okay, so these are the various glial cells in peripheral nervous system: satellite cells, Schwann cells. How about the glial cells in the central nervous system? Oligodendrocyte, astrocyte, microglia, ependymal cells. Okay, so what are the types of neuroglia? First of all, on our list we have the microglia. We say that a microglia. This is a microglia. Okay, this is a microglia. We say that the microglia are specialized macrophages capable of phagocytosis that protects what neurons of the central nervous system. And also they are derived from the hematopoietic precursors rather than ectodermal tissue. And these cells are found in all regions of the brain and spinal cord. Okay, microglia cells are small related to the are small relative to macroglia cells. Note the difference between microglia and macroglia. So now with changing shapes and oblong nuclei. Okay, you see the nuclei is oblong and this shape is prone to change. Okay, so they are mobile within the brain and multiply when the brain is damaged. Okay, then how about the macroglia? We said that the most abundant type of macroglia cell in the cortex is the astrocyte. Okay, also known as the astroglia. Now, astrocyte is maybe it's like a star shape. Okay, so the astrocyte have numerous projections that anchor neurons to their blood supply. Okay, they regulate the external chemical environment of neurons by removing excess ions, notably potassium, and recycling neurotransmitters released during synaptic transmission. Now, they are predominantly building blocks of the blood-brain barrier too. Okay, so now in general, there are two types of astrocytes. Okay, we have the protoplasmic and the fibrous. Okay, so they are similar in function but distinct in morphology and distribution. Okay, and further, the protoplasmic astrocyte have short, thick, highly branched processes. Okay, and are typically found in the gray matter. The fibrous astrocyte have long, thin, less branched processes. Okay, and are commonly found in white matter. Okay, so these are the astrocytes that we are talking about, and they are types of macroglia. Okay, and we have two types: we have the fibrous and the protoplasmic. This page explains where the two types are predominantly found. Okay, then next we have the oligodendrocytes, and these are cells that coat the axons in the central nervous system with their cell membrane forming a myelin, okay, producing the myelin sheath. The myelin sheath provides insulation to the axon and increases velocity impulse conduction. Okay. Next we have the ependymal cells. The ependymal cells, also named ependymocytes, they line the cavities of the central nervous system and make up the walls of the ventricles. Now, these cells create and secrete cerebrospinal fluid and beat their cilia to help circulate the cerebrospinal fluid and make up the blood cerebrospinal fluid barrier. Okay, then this is followed by the Schwann cells. Schwann cells are similar in function to the oligodendrocyte. They provide myelination to axons in the peripheral nervous system. They also have phagocytic activity, and they clear cellular debris that allows for regrowth of peripheral nervous system neurons. Okay, then next we'll be moving to the cellular cells. The cellular cells are small cells that surround neurons in sensory, sympathetic, and parasympathetic ganglia, and this help regulate the external chemical environment of the neurons. Okay, um, the sensory, okay, the sensory, right? So these are notes on glial cells, okay, um, types of glial cells and their functions. Okay, see you guys in the next tutorial as we continue talking on excitable tissues, and bye for now.